The next class of organic molecules that we're going to be talking about is proteins. Now, the structure of proteins, proteins are made up of amino acids, and we talked about acids already. And so if you look at, again, look at the sheet that I gave you, because for video number 6 is the area you want to be looking at. The structure of an amino acid, you should recognize this carbon doubly bonded to an oxygen and singly bonded to another oxygen and then a hydrogen. So you should see this acid group on the end of this amino acid. So here's the acid part. On the other end, you have a nitrogen with 2 hydrogens attached to it. And this is called an amino group. You don't need to be able to recognize it, but I'm just telling you that that's what it is, because that's where the name amino acid comes from. Now, this amino acid is not very big, but remember what this R stands for. That R just means it's attached to something else. And that something else could be one atom, it could be a huge number of atoms, it could be a whole series of carbon rings, it could be a really, really long string of carbon atoms. So this is just a something else. And so here I have an amino acid. Over here I have another amino acid. And just the example that I found on this one, in place of going from left to right, it happens to be going from top to bottom, so don't let that confuse you. Okay, so just like we had dehydration reactions with carbohydrates, and we have them with lipids, we have them again with proteins. And so I'm still going to be looking for that OH on one atom and the H on another one. And then we take this OH here and this H here, we suck that water out of there. And once I do that, then these guys are looking for something else to bond to. And so the carbon and the nitrogen are now bonded to each other. And I now have, this is called a peptide bond, which is the kind of bond, and it's right here between this carbon and nitrogen. So here's your peptide bond. And peptide bonds are what bond together amino acids to make polypeptides or proteins. Okay, now, in order to make a protein, I have to link together at least 124 amino acids. That's the smallest protein that exists. And so my whiteboard isn't nearly large enough to put together that many amino acids. So you can only imagine how big these molecules are. There are 20 different amino acids that link together to make proteins. And so these molecules are very, very big. The property of the protein is determined by what type of amino acids are linked together. Remember, I just told you there are 20 different kinds. So it's determined by what type are linked together, how many are linked together, and what order they are linked in. And there is a really good page in your book entitled Creation Connection, and it's on page 73. And she goes through a really good explanation there of how impossible it would be for all of the proteins that are necessary for life to just randomly come about, which really points to the fact that we do have an almighty, all-knowledgeable creator who has put all of this together. So make sure that you read through that, because it kind of makes your head explode as to the possibility that even people could even think that this could happen by some sort of random reactions that could make these amino acids and these proteins like this. Okay, so make sure you look at that. Then in your organic molecule chart, we'll get that filled out here. The organic molecule is the protein. The building blocks are amino acids. You can get these in your diet from things like meat and eggs. How is it used in our bodies? It's used to make hair. It's used to make muscle. It's used for long-term nutrient storage, defense from microorganisms, and it's also used to carry messages. And a special class of proteins is an enzyme. Now, what is an enzyme for? Well, there are a lot of different chemical reactions that have to happen in our bodies. And some of those reactions would be painstakingly slow, and they would be so slow that we would have trouble surviving if we weren't able to somehow kick up the speed of that reaction taking place. Now, the easiest way to speed up a reaction is to raise the temperature. But if you raise the temperature too high, then that can kill your organism. So there has to be another way to increase the speed of a reaction without getting the temperature to such heights that it would end up causing death. So an enzyme is a specific type of protein that's used to enable a reaction to run at a lower temperature. And what it does is it is a special type of molecule that enables the molecules that it's acting on to bond to it in a special way so that it will enable the molecule to be broken down. So as we've been talking about all these dehydration reactions that will make molecules bigger, our bodies have to be able to break the molecules down in order to use them for other things. Just like if you think about your Legos, if you take your Legos and you build them all together, in order then to build something else, you have to take those blocks apart. Well, it's the same way with our bodies. We have building blocks that make up the protein in the meat that we eat or the eggs that we eat. And then our bodies then have to take those and break them down to separate the amino acids out so our bodies can reform those amino acids into the type of proteins that it needs. So if you look at the picture underneath the reaction for video number 6, you see a representation of what it looks like for an enzyme. The enzyme is the gray piece that is there, and what is on the top is a polysaccharide that they are going to try, and I mean that could be what this is, a polysaccharide that they're going to try to break apart, and it is hooked together. So it has special places on the enzyme where this molecule is able to come in and fit exactly, the shape fits just the way it's supposed to. Then the enzyme acts on it, and if you would add water to it, if this would happen to be a polysaccharide, you add water to it, and then you get that polysaccharide broken down again into monosaccharides, and then they're split apart. Now you can see that the enzyme was necessary there for the reaction to take place, but it didn't get used up. And so that enzyme can be used over and over and over again to keep breaking down the types of molecules that your body would need to break down in it. This is called a lock and key theory, because just like a key fits only one way into a lock, these other molecules will fit into the enzyme, and then they can use that enzyme then to be broken apart so the body can use the building blocks in other ways. Now, even though enzymes are very, very critical to the body, it is also true that it is pretty easy to destroy them. And they can often get destroyed if the pH gets out of balance or if there's too much heat. And when that happens, that's called denaturation, and it basically just means that your enzyme gets destroyed and is no longer functional. So they can't be used in places of high heat, or they can't be used in places where you've got a pH that's not compatible with their normal functioning levels. Okay, so that is proteins, and then we will have just one more organic molecule to go.